Hello, welcome to another video from the conscientious biologist, Ben Gallagher. This one's on meiosis, but I've included a recap of mitosis and a comparison of the two, because that's quite often how they ask you in the exam. They'll ask you for the differences between mitosis and meiosis. Now, this is a really complicated process. So this is one of the more difficult videos. So really give it your full attention. If you get to the end and you're still not sure, watch it again, make notes as you go along, because this is such a key process. And this is absolutely vital for understanding the genetic inheritance lessons for how genes get passed from one to another. Now, I am going to go into it in quite a lot of detail. At the end of this video, I'm going to give you summaries. If you've really struggled with the understanding, just focus on the summaries. But it's important you try and understand the complexities. This is from the GCSE specification, and as always, please do subscribe to my YouTube channel if you haven't done so already. Thank you. So let's start off with our mitosis refresher. Now, mitosis is normal cell division that's happening all over your body to make copies of cells, to make clones, okay? Mitosis is for growth and repair. Growth when you just need more cells to get bigger, and repair when you need to replace cells that have been damaged. So we start off in just the normal start of any cell's life in that it's quite small. It has single chromosomes inside it, a full set of single chromosomes though. And it needs to grow and double and get bigger so it can divide. Now, if it's going to divide into two, each new cell is going to need a new full set of DNA. So the DNA that was there at the start, those single chromosomes, need to divide to become double chromosomes. Now, plus, of course, during that stage, it's doing all its normal metabolism and life process that it would be doing. This is classed as stage one of a cell's life. It doubles in size, make copies of all its DNA. Then it's going to enter the start of mitosis, where the nucleus breaks down and those double chromosomes float off through the cell. They float off, and each double one is grabbed from both sides within the cell, and it's pulled apart to break apart the two single chromosomes from the double, to break them apart, to pull them to either end. Now remember, the two halves of a double chromosome are identical to each other. So when you pull them to either end and repackage them in a nucleus, each of those two nuclei will contain identical sets of DNA. Now, if you weren't sure about what I'm talking about with the double and single chromosomes thing, let's just take a little bit of time recapping that. So this is a double chromosome. Each one of those strands is an identical copy of the other one. The cell started off with just one. Because it needed to make two for when the cell divides into two, it made itself an identical copy. Okay, but so a double chromosome is basically two single chromosomes. But because they're identical to each other, you still just class it as a chromosome, a set of that particular set of instructions. Now, what's going on in stage two is that double chromosome gets broken apart at this point where they're held together, and they get pulled to opposite ends of the cell. Once they're there, they get repackaged. Okay, so that's what we're talking about with the single double chromosome thing. Let's get rid of those for now though. Nucleus forms around each set, and then of course the cell pinches in the middle, divides into the two cells. Both of those cells here, look at the very end of the diagram, they are absolutely identical to the one that's over on the end there. Okay, exactly the same number of and type of and instructions and genes and DNA. They are identical cells. So that's stage three. And stage 2 and 3 together, making those cells with identical DNA, that's mitosis. And mitosis, as you remember, is for growth and repair. So take a screenshot of this to add to your notes for where you're doing meiosis. You've got this as a reminder. But if that was too quick and you're not sure, go back to the video of mitosis that's in the cell biology playlist right back near the start of phase 1. Okay, so let's get started with meiosis then. Now, the really important thing with meiosis is it's specifically for the production of gametes. So it only happens in very specific areas of the body, in the testes and the ovaries in humans, to make sperm and to make eggs. Okay, so mitosis is everywhere, meiosis only in very limited places to make those sperm and ova. The first stage is exactly the same as we saw in mitosis. The cell's got to double in size so it can divide. And of course, as it doubles in size, it needs to replicate that DNA and turn its double chromosomes into single chromosomes. Now what it does as it enters meiosis, is the nucleus breaks down to release the chromosomes, just like we saw at the start of mitosis. But you'll notice something different here in my diagram. The chromosomes haven't all floated off individually. They formed these partners. They found a near identical partner to them that's called a homologous pair. Okay, now to explain a little bit more about that, if I put these two over there and we call them a homologous pair, just like you've seen paired up in the diagrams up there. Note though, this is where some people get confused. It's a homologous pair of double chromosomes. So people go, well, hang on, how can it be a pair and a double? Is it double or is it a pair? It's because each chromosome is a double because it's made of two single chromosomes, but there's two of those double chromosomes, so they make a pair. Now, the really important thing here is that the two parts of a double chromosome, they're identical. Remember, they were made from each other. They're literally an identical copy that's made so that you've got two copies to split between the cells that you're going to divide into. So those two are identical, and of course on the other double chromosome, they're identical. But this is the important bit. The two on different ones in the homologous pair are almost identical, virtually the same. And how they're almost identical is that they can have the same genes for the same jobs. In other words, the proteins that are produced from them do the same job. They might have, like, all four of the pieces of DNA that you can see in that pair of doubles, they might all have the insulin gene. All of them will have the gene for insulin. So you can say, well, therefore they're identical. If they all have the same genes, they're identical. But this is where it's different, because not all genes are the same. You can have different versions of a gene. Now, to give you a really obvious example of that, they might all have the gene for eye color. They might all have the gene that makes the protein that sits in the iris of your eye that decides whether your eyes are brown or blue or green or whatever. Okay, so they all have the eye color gene. However, some of them might have the green eye color gene that makes the green protein. Some of them might have the brown one. Some of them might have the blue one. So they can have versions of the gene. So it does the same function, but it might do it in a slightly different way. Going back to the insulin one, the homologous pair nearest to me, both its parts might have a functional insulin gene. On the far one, both its parts might have a non-functional insulin gene, which would cause diabetes. So they've both got an insulin gene, but one works and one doesn't. So they're different versions of the same gene. That's what we're talking about with homologous pairs. Let's carry on with the process. Oh, sorry, quickly. Where do these come from? Why have you got two? Is because you got one from your mum and you got one from your dad. So this is where it gets really important, in that you actually have twice as much DNA as you actually need. Your mum gave you a full set and your dad gave you a full set, because a full set of human instructions is actually just 23 chromosomes and we all have 46. So my dad gave me 23, a full set of human instructions carrying every gene for every protein that's needed for making a human. My mum also gave me 23, covering every gene for every protein that's needed for making a human. So I get two full sets, hence why half my chromosomes are maternal from my mother, half are paternal from my father. So that's why I've got homologous pairs, because they come from both of my parents. So you've got a full set from each parent, giving you 46 in total. That's why chromosomes have a near identical pair, because you've got your maternal one and your paternal one. And meiosis is to split that double set back into a single set. So what you pass on to your offspring is back to a single set of human instructions rather than the double set which all of us hold. That's new information. That might be a bit weird to some of you, that you actually hold 2 humans worth of DNA. But that's really important for your variation, because it gives you some choice. You can pick and choose from which genes from your mum, which genes from your dad, which is why you're a mixture of both. But you've got one of each gene from each parent. So you've got 2 copies of everything. Let's just get rid of that for now and go back to our process of meiosis. So if we've still got that first stage where it grows and it replicates its DNA, but now the DNA has been released, that the chromosomes know how to find their homologous partner. Now what happens is the pairs get pulled apart, not each double chromosome. You're not tearing a double chromosome apart like you did in mitosis. You're pulling one of the double chromosomes down and the other one will go up. And this happens with each one of the pairs, as you can see. So what actually gets pulled to each end are the double chromosomes. So that's quite different to what you saw in mitosis. We'll compare them in a second. And of course, a new nucleus is going to form around each one of those. The cell's going to pinch in the middle, and it's going to divide into the two cells. So the diagram looks very similar to what we saw in mitosis, but what's in the nuclei is different. And if I just get rid of the start bit and pull up that end bit to compare with mitosis, so you can see there that in mitosis, you pulled apart the double chromosomes back into their singles and repackaged them. So if you look at the cells nearest to me here, each one's got 4 single chromosomes. If you look at the ones up above me, because you pulled apart the homologous pairs but left the double chromosomes intact, each one of these cells up there has now got 2 double chromosomes. But of course, 2 double chromosomes technically is made of 4 single chromosomes. Now that is important because in this one, all the chromosomes in the 2 cells, they're identical in the 2 cells. The 4 in that one are identical to the 4 that are in this one. Whereas here, in one of the cells, you've got the maternal version, and in the other one, you've got the paternal version. And same for each of all the pairs. So you've shuffled here your mum and dad's DNA. In making your gametes, you've reshuffled the ratio of your parents' DNA. And that's what's really important. But this is only the first stage of mitosis. So let's look at what happens next. So let's pull that whole diagram back up that we've just talked about, where we end up with these cells up here that contain double chromosomes still. But have a quick think. How many double chromosomes would be in each one of these cells? If we started off with 46 chromosomes, way over there in that first one, there would have been 46 single chromosomes in that one. Then the cell grew and replicated its DNA. Now we've got 46 double chromosomes. The double chromosomes all found their partners, their homologous pair, and then got pulled apart. So how many would be in these ones? It's 23. Each one of those cells would contain 23 double chromosomes because the 46 doubles found their partners. So you have 23 homologous pairs. Each of the 23 homologous pairs got pulled apart. So 23 double chromosomes went up, 23 went down. The cell pinched. So these cells contain 23 double chromosomes. Now, if I take the bottom one and we look at the next stage, what happens now is basically exactly the same as mitosis. The nucleus breaks down, double chromosomes are released, the double chromosomes get broken in half at that middle point, just like we saw on the first slide in the recap of mitosis. They get pulled apart back into their single chromosomes, which get pulled to opposite ends. A nucleus can reform around those. The cell's going to pinch, it's going to divide into 2. Look at those ones down the end there now. Those ones have only got, on my diagram, each one's only got 2 single chromosomes. Compare that to the cell at the very start of the process right up in the corner, that had 4 single chromosomes. So we've exactly halved what we originally had. Of course, going by the actual numbers, that original one in the far corner had 46 single chromosomes. That replicated into 46 double chromosomes. At the top up there, they divided into 23 double chromosomes. Then the double chromosomes got broken in half to the ones over there that now have 23 single chromosomes. But of course, that didn't just happen with the one cell on the bottom there. That top one did this exact same process as well. So this is now showing the complete diagram for the mitosis that would happen to that one original cell over there. We end up with 4 gametes being produced. And those gametes now contain half of the quantity of DNA. You can just look at my diagram. They've got 2, they've got 4, so you've halved it. But this is the really important bit. All 4 of them are genetically different because they've got different ratios of the maternal and paternal DNA. So they've got a different mix of the versions of the genes. The top one might have more paternal genes than maternal. So someone who grew from that gamete, or the 50% that came from that gamete, would be more paternal than maternal, and so on as you go down. We'll explore more of that concept when we look at the genetic inheritance video. But this page covers all of the information for what happens in meiosis, okay, or all of it diagrammatically. Now, I said at the start, this is a big, complex, long process. So if you need to watch it again, or take a screenshot of this and print it out and write notes all around it, do that, whatever's easiest for you. But this is a big, complex process, but it's really important and vitally important for your understanding of genetics, which we'll go on to in a couple of lessons' time. Okay, now that's all really complex stuff. And if you're aiming for kind of the top levels, level 7, 8 or 9, or if you want to make sure that you have a really good understanding of genetics and inheritance when we come to this, then you really need to make a big effort to have learned everything I've covered in the video so far. However, if you think that's just really too much of a challenge and it's too difficult, that's absolutely fine. I'm going to give you a simplification and a summary now. And this is just for human cells. So if I put up my simplification for mitosis now, this is the same simplification I used at the end of the mitosis video back in phase 1. You start with a cell with 46 single chromosomes. That cell grows, replicates its DNA to make 46 double chromosomes. It takes each one and makes an identical copy which stick together. That's your double chromosome. The double chromosomes are then split apart. So you've got 46 here, 46 there, making your 2 new cells. Very straightforward. Let's do the same simplification for meiosis. You start with 46 singles, that becomes 46 doubles. Exactly the same as phase 1, stage 1 that we saw in mitosis. Now it gets different now. In meiosis 1, that's the first division, the first line that we looked at previously, you separate out the homologous pairs. So you still keep double chromosomes. You don't break the double chromosomes up. You just separate them from each other. So you go to 23 double chromosomes in each. Then in meiosis 2, you separate those into 23 single chromosomes. So if you look at the numbers, you've got exactly half of what you originally started with. And the really important thing is that those 4 cells show variation from each other, whereas the 2 cells here are identical copies of that one. So mitosis, identical copy to the original. Meiosis, half the quantity, and they're genetically distinct. Now, while we've got this summary up, I'm just going to add a couple of new words on here, which are essential for all of you to know, whatever level you're at, because they do get used in the exams. And they're the words diploid and haploid. Now, I said before that you contain a double set. You've got a full set of DNA from your mum and a full set from your dad, and a full set's only 23 chromosomes. So technically, we've got 2 lots of the numbers of genes required. So when it's 2 lots of the number, n for number, we say 2n, and we call that diploid. Okay, it's referred to as the ploidy number. The ploidy number is how many sets of DNA do you have. So in that one, di quite often means 2, diploid meaning 2 full sets. So when that multiplies up to 46 double chromosomes, it doesn't become 4 sets, because you've still got your mum set and your dad set. Yeah, all right, you've got 2 copies of each one, but you've still only got 2 different sets. Okay, it's like if I had 2 copies of the Wind and the Willows book. Okay, if I had 2 copies of the same book, and people said, oh, have you got Wind and the Willows? Yeah. Good. Have you got any other books? I wouldn't go, yeah, I've got another Wind and the Willows. You go, no, I've just got Wind and the Willows. I've got it twice, but I've just got that one story. Same here. You've got diploid because it's 1 set from your mum, 1 set from your dad, 1 version of instructions. So when you multiply that up into the double chromosomes, it's still diploid. Even though you've now got twice the amount, you've technically got 4 sets. It's only 2 different sets. So that's diploid. However, when you half it down, you've now got half haploid. 1 set, 1 type, 1 version, 1 copy of the book if you want. So it's n. When you break it down right to the bottom, those are still haploid. They're still just n. So diploid 2n, full set of DNA, technically double, and so on. If we apply those same terms to mitosis, they're all just diploid. because they're all identical copies of the originals. So diploid and haploid, terms that you just have to know. Make sure you do. Okay, so quite a few summary points here that we need to look at, and let's do the summary points for both mitosis and meiosis. So mitosis, really important, makes two genetically identical versions, and it's for growth and repair. And mitosis is just part of the cell cycle, so it just keeps going round and round. That cell gets bigger, divides, each one of those gets bigger, divides, and so on and so on. It keeps going round and round and round. Meiosis isn't a cycle. Once you've got the cells in meiosis, they're either used or they die, okay? But in mitosis, the original cell and the two cells it divides into a diploid. Now, meiosis creates gametes, which are sex cells. It's got 2 rounds of divisions you can see here, which means 4 gametes can be created from one cell, not just 2 cells in mitosis. Meiosis halves the chromosome number. That makes the gametes haploid. And meiosis shuffles the maternal and paternal versions of the genes to create variation. This is the most important slide in terms of information from this process. Make sure you've got a screenshot of this one, take a screenshot of the last slide, and ideally a screenshot of the full process, and add your own notes to those. Now, I'm not expecting you to have found this video easy or straightforward. It may have just confused you even more. This is probably one of the most complex topics of the GCSE. So go back and watch it again if you need to. Make your own notes, look it up in your own textbooks. Obviously, your own teacher is going to be teaching you this in school as well. But it's a key topic. From here, go on to asexual reproduction. This is still within the genetics and inheritance playlist. After that, you'll go to sexual reproduction, and this video, the asexual reproduction video and the sexual reproduction video, very much are the baselines that lead on to genetics and genetic inheritance. If you haven't done so already, please do subscribe to my YouTube channel. It really helps me out having more subscribers. Like this video if you find it useful, and there's loads of extra information on my Facebook page, so go and have a look there if you're on Facebook. Thank you.